Okay. You hear me only once, I hope? Okay, that's good. Well, I would like to thank first Michiel and the organizers for inviting me, basically to speak about the thing I like most, which is flatworms and taxonomy of flatworms. Why I think it's so interesting, because it's one of the, and I will explain it a bit later too, it's one of the taxa which are least known in the marine realm, but also in freshwater and even less in semi-terrestrial or terrestrial environments. So they are quite obvious, well, quite rare, or rarely studied, I should say. And in that respect, they are not different from many other taxa, maybe, because when you see things like this is from the Census of Marine Life, you see that still quite a few animals need to be described. They estimated like about 953,000 animal species already described and 7,700,000 to be described still. So we have quite some work to do being alpha taxonomists. And it's important, for one thing, if you look at species diversity, it's nothing new to you, I think. They're pivotal. Knowing the species diversity in every habitat is pivotal, just because it's so important and is in between what we call genetic diversity, which underlies it, and ecosystem diversity, which we will hear of later this day, I think. And they all interact with each other. But in the middle of this is the species diversity, which is maybe very accessible still, but very, very, very badly known. And even in a time, everybody knows at this time that we are in a phase of mass extinction. Some people even say we are in the 6th phase of mass extinction of biodiversity in the world. And you see these graphs, which you can find everywhere, where you see that from 1900 on, from the 20th century on, species get extinct in an enormous pace at this moment. And this is a conservative estimation. This is a more daring estimation, I should say. But we are really in a phase where many species face extinction. And knowing that we don't know much about just the diversity of life on Earth and seeing these type of graphs, it's quite uncomfortable, to say the least. And when you say these graphs, you see the mammals and the birds and the vertebrates and still other vertebrates. But where are the invertebrates? That's something we really don't see much about. And then you see these tables appearing. This is from 2003, but things didn't change much since then. You see all these counts of number of described species and number of species evaluated and number of threatened species and so on and so forth, with very detailed numbers for mammals, birds, reptiles, amphibians, fishes. And then you have the invertebrates with insects, mollusks, crustaceans. And then you have this very strange thing, others. Which means that apart from the arthropods, mollusks, and crustaceans, basically what these things say, for all the other invertebrates, we don't know anything at all, or very little at least. But if you have to look at these others, here the animals, well, larger taxa, which we used to call phyla, are summed up. Then the others is basically this. So for about 31 of the 34 larger phyla we have, this is lumped into others. Why? Just because we don't know much about them. And in the past we saw many of these initiatives, which many of you know, I think all of you know, such as the World Register of Marine Species, such as the Catalogue of Life, the Freshwater Diversity, Animal Diversity Assessment, which at least tried to bring together all knowledge on species diversity in the marine realm, in the freshwater realm, to get an idea what is there. And based on these, I should say, especially for worms now, excellent data sets, there were publications which gave number of species and an estimation. This is based on this paper by Appeltans and a huge number of co-authors who gave input to this, who tried to assess the number of marine species and an estimation of how many species still are there to be described. And if you see the number of, these are the 10 most species-rich taxa that came out of this count. And if you have a look at the flatworms, this is one of the least known taxa there. Although the number of species is quite huge. And this is one of the reasons why our group already for many years, I think now since 1976 more or less, I'm doing research since 1995 on diversity of flatworms. And that's why we try to at least help to get known the diversity of flatworms worldwide in this respect. Now, for you who forgot or didn't know, what are flatworms? Flatworms are an enormously large group, species rich, with about 13,500 parasitic species, which we know as the neodermata. These are the tapeworms, the flukes, the monogeneans. Rather well studied, although parasitologists will not agree with me that they are well studied, but much better studied than the free living ones. And they're, well, a monophyletic group, as we call it. They are an evolutionary unit. They have one point of origin. All the other ones are known as the turbellaria. And if you see, there are only 5,200 species known, more or less, although they are extremely diverse. And I write it like this. This is called turbellaria. It's not a taxonomical unit anymore. This is a bit technical. But if we see at the evolutionary history of the flatworms, or the cladogram as we call it, these are the flatworms, one big group with one ancestor, so a monophyletic group. But if we subtract the neodermatans, then we see that the turbellaria, which is all the rest, is not monophyletic. So it's a paraphyletic group. It does, but this is a bit technical maybe. Now, if we have a look at the turbellaria, many people know them as small animals, but it's not entirely true. You have the microturbellarians, microscopical ones, which range everything between 0.5 millimeter to 3 millimeters, more or less. And then you have the medium-sized ones, which all of them of you know maybe as the planarians, which are these freshwater animals you can find in your ponds in the garden and everything. And then you have the very large one, up to 50 centimeters, which are terrestrial ones, more often also the triaglets, land planarians, and so forth. My research, or our research in our group, basically is on microturbellarians, so the small ones. There are a number of microturbellarians, as you see. These are the larger groups of them. I'm not going into detail. But here are the species numbers of this. So there are quite a number of them. 110 of catanelids, 109 macrostomids, 1700 rhabdoseals. And if you see these species numbers, and this is quite discomforting. I mean, there are, and I will show it to you later, but many species are not known. We guess about 10%, more or less, of the world's species are known. And these are the taxonomists working on them. So in total we have 4 worldwide. I mean, this is lately a little bit exaggerated because it's 4 PIs, so it's 4 labs. So maybe you can say we have, like, at this moment 4 PIs who have as main research flatworm taxonomy and phylogeny. This is quite discomforting. And if I didn't put the age of the taxonomists there, but it's they're in the phase of extinction too, I should say. Yeah. So we have a huge task to do. But if you see why are they basically important, as I said, so the microturbellarians occur basically everywhere. Everybody, well, I think most of you know they occur in marine sediments. They occur in sandy beaches, in algae, rock pools, and so forth and so on. But they also occur in freshwater, in all types of ponds, going from very pure mountain streams to very ugly, marshy ponds and stuff like that. And they even occur limnoterrestrial, or terrestrial you can call it, in mosses, in leaf litter, in woods, and this is a very, very unknown fauna. Nobody knows about them. For instance, nobody ever looked at terrestrial flatworms in rainforests, for instance. We don't know anything about them. Nothing. When you go to see at the marine interstitial microturbellarians, so microturbellarians living in sandy beaches in the interstitial space, I made a quick count, and there's a very rough estimate that only 10% is described. Yet these turbellarians are one of the major components of these groups. Now I should say nematodes and harpacticoids are not very well known either. But I always have well, as to species diversity is more or less the same. But flatworms are not marginal in these areas. They occur quite often and quite numerous. This is seen in a very old study, well, very old, end of the 1980s, by Philip Yuck, who gave me this graph, where you see the turbellarians in purple, and this is the percentage of occurrence, of abundance. Then you see the nematodes are very abundant always. the harpacticoids, so the crustaceans, and the turbellarians. These are the three most common groups at our coast, because this is Hasselt, Oostende, Mariakerke, Bredene, Knokke, and the Zwin. So they occur quite often. And this was confirmed by a molecular study, an environmental sequencing study by, I think most of you know, Fonseca et al. 2010, which was a kind of a groundbreaking publication. I'm not going into the details, but the flatworms in all the stations they did, I think it was about 9 stations in Scotland, where they just extracted all DNA. You see the flatworms coming out second in all of these samples, as the second most abundance. As well, also here you see it in number of OCTUs, which you can say, well, putative species, more or less. So flatworms, and this was confirmed in studies after that too. So the flatworms are not a marginal group within beach ecosystems, at least, as far as we know. Now, what role do they play in ecosystems? We don't know anything. You're going to hear me say that a lot during this talk. We don't know much about it. The only thing we know, and this is just from some things of our research, what do you find is predators, at least. You see here, this is the front of a, well, the front end of a flatworm with large jaws, which they catch nematodes. Well, I saw everything basically in flatworms. They eat nematodes, they eat tardigrades, they eat copepods, they eat themselves. I mean, everything that's coming to reach, they eat, basically. So they could be, I think they could be called top predators in the interstitial marine habitats. But you also have algivores, animals who eat algae, like you find the gut filled with algae at that point. Diatoms for a lot of diatoms. You have even flatworms with symbiotic algae. Now somebody in Canada is doing research to that, to see how the relation is between the symbiotic algae and the flatworm, because they acquire it after hatching. So when they hatch, they don't have it. And then you have symbionts, mostly of echinoderms and mollusks. And they all live together in one beach. Because they are so diverse in feeding, I think that's why, but we don't know really. But the diversity of species in one single sample of flatworms can be high. I mean, 35 species of rhabdoseals alone, which is one group, in one beach is not uncommon. And it's possibly because they avoid competition in this way just by going in all different food sources. But this is just gut feeling. I mean, nobody ever did research on that. If we go have a look, where did we study flatworms already? And this is from, I extracted it from OBIS, only for the rhabdoseals, which is 1,700 species. I will come to these rhabdoseals later on too. But as you see, the numbers are like here. This is 100 to 200 species described. This is Europe. But for many coasts, as you see, nothing ever was done. So we don't know anything about biogeography, to be very honest. It's very, very difficult. For freshwater, it's the same. As I said, all of Asia, there is nothing going on. Nobody knows what's there. So this makes research quite difficult. And not only that, and of course when you have a look at it, these are some numbers I had. We went to Sardinia. Well, I was invited to some workshops lasting more or less 2 weeks, 2 to 3 weeks in all cases. There was one in Sardinia, well, 2 in Sardinia basically, Lanzarote one, in Panama there were 2. But in each of these sampling campaigns we found about 75 species of rhabdoseals only. And then you see the species which are new to science. This was Sardinia, which is considered one of the best known places of the world, together with Western European coasts, which mean that half of the species we find there are still new to science. So there is quite a lot of work for the 4 taxonomies still alive in the world. So what is the difficulty about studying flatworms? Not taking the samples. The only thing you need is a small veil and a shovel. That's the only thing you need. And you go out and you take samples. If you change from habitat, it can be more daring. So if you want to see, and the species composition really differs, so we sample as much habitats as possible. The one you see here is Marco Corini Galetti, the only specialist of proceriates still in the world, one of the 4 or the 5 we still have. So if he didn't survive, there was 25% of the taxonomists worldwide dead. I mean, quite a risk we took there, sending him out. But he's the oldest one, so that's... So this is basically the samples we have, and they don't need much, how do you call it, taking care of. You just put them in bags or put them in vials. You take them to the lab or to where you're working. That's it. I mean, it's not more than this. There the samples are. And then you have to process them, which is a very easy process. I won't go into details, but it's very easy, both in marine and freshwater habitats. It's no problem at all. You don't need fancy stuff. You just rinse them, separate, extract the animals from it, which is easy. You don't need fancy labs or whatever. You can do it everywhere, in kitchens, in outside. But then the problem starts. Because flatworms are soft-bodied animals, you can't fix the sample and study them afterwards. You have to study them on site where you are, alive. Which means that you take every specimen, put it under on a slide, and study it. Taking pictures, drawing it by hand. If you think it's interesting, you have to fix it with lactophenol. So it takes you about, if it's a known species, 10 minutes to a quarter of an hour. If it's a new species or even a new genus, it can take like 3/4 of an hour to an hour to study it alive. Every specimen. I mean, that's something which makes it extremely difficult. When you go out sampling, people always ask me, well, can I take samples I'm going to this or there, can I take samples from you? No, you can't. Because I have to study them alive. And that's a difficulty. And these are the results. This is a sketch of a new species we had from Australia when we were there. So I know the genus, but the species is new. And then these are the pictures of the copulatory organs, which are quite difficult to draw, I have to say. And the pictures and everything. So this takes time. It's very, very time consuming. It's not easy to, it's not like belt work. It's a problem. Moreover, when it's a new genus at least, or a very badly known genus, the species are, you need sections. So we make sections of 4 micrometers, and then you put everything together by hand, getting to these type of reconstructions. And to be honest, when you're very experienced, you make a reconstruction like that in, if the sections are alright, a week, 2 weeks time, full-time work. So it's very, very time consuming, and that's why many people don't want to get into flatworm taxonomy, or alpha taxonomy, I should say. So very little is known, it's very time consuming, with very few taxonomists still alive, but yet we were able to do at least some interesting work, I think. Well, at least I think it's interesting. I hope you agree with me after this. But anyway, we focused on the Rhabdocela. Why the rhabdoceles? It's many, for many reasons basically. It's a very diverse group. As here you have many examples, but they occur in all marine environments, they occur in all freshwater environments, they occur in semi-terrestrial environments. They are symbionts there, living within echinoderms, within mollusks. So it's quite a group with a lot of diversity, morphologically as well as ecologically, and that's why we focus on them. Moreover, if you see the number of species, 1/3 of the marine species is a rhabdocele. Almost half of the freshwater known turbellarians are rhabdoceles. 60 limnoterrestrial species are known, of 1,000, but basically 900 of these 1,000 are triclets. So let's say 100 of the microturbellarian terrestrial species, so 60% of the microturbellarians are terrestrial rhabdoceles. And then you have in total almost 1/3 of the turbellarian species known as rhabdocele. So that's why we study them. And traditionally there was until 2006, so almost for 70 years, this classical taxonomy was used, where you had 3 big groups, Calyptorhynchia with a proboscis, and then 2 groups without this proboscis, Typhloplanoida and Dahlialuida. So in 2006 we did 5 years before 2006 we started already. We did a study on these animals. And the first thing we saw is that these groups together form one bunch. So this is the phylogeny we had with the Calyptorhynchs, with the proboscis, and the other ones, 2 clear monophyletic groups, sister groups. This is not very important. when we had a look at these ones, we're not now going to have a look into the Dali-Tiflo planets. We saw already that morphologically speaking, the Dalialids are not related to each other. But what's more importantly, when we put habitat on this, then we see that we have clear blocks of freshwater taxa, which are morphologically extremely diverse. Sorry, this is marine taxa and freshwater taxa, with a large polytomere. Basically, we didn't know at that time how they were related to each other. So this was quite interesting to dig into, which we did. So you see, this was 2006. In 2012, we had sampled many more species. And this was a sampling effort which was huge, I should say, worldwide, with heaps of new species we found and many we described. But what came out of this is that there is, if we have to look at the habitat, within these rhabdoseals, all freshwater rhabdoseals form one and a single clade. So apparently, from the marine environment, there was one invasion into freshwater and then a huge adaptive radiation within the freshwater. And morphologically speaking, it went across morphology. Morphology is in the bigger taxonomy of the rhabdoseals is basically not very important, apparently. A lot of convergences. So there was this one invasion of the freshwater. And if we have a look at this freshwater clade, don't look at the names or anything, but then you see within that there was one clade which became symbiotic, possibly one, possibly two re-invasions of the marine water. But the single colonization event, that was really strange to us. So there was a first single colonization of the fresh water. But then we had a look at these terrestrial flatworms nobody know anything about. We had some collected in Belgium. And then you see they are scattered around. So we tried to figure out what happened here. Is this the classical hypothesis was you had marine, and you went from marine to freshwater, and then freshwater to terrestrial. That was the idea already since 150 years, basically, from Grave on. Then we see these limnoterrestrial or terrestrial rhabdoseals scattered around within this freshwater clade. So were there more invasions of the terrestrial realm? What is going on? We didn't know. So we investigated that. This is kind of the environments they lived in. They always say they are associated with water, but we know now they can survive in desiccated moss for months. More or less doing what the tardigrades do, so they encapsulate and always lay a couple of eggs before they do so. And they can live for weeks, even months encapsulated, the worms, the adult worms. When you put them into water, they come out of the cyst and are laying eggs in 3 or 4 days. So it goes very quickly. So this type of animals we started to collect, which was quite funny. We just get out of the university, took our first samples and had a new genus. I mean, that's how badly known they are. So we were sampling mostly in Western Europe. And some in North America too. And then you see of the rhabdoseals, 2 semi-terrestrials are in this clade, and 46 are in this clade. In the meantime, we have described like 10 or 15 new species, so we are 60 now. But we are going to focus on this one first. So if you have the clade, and we did a Bayesian trait analysis, this is just to see what the ancestral habitat was, don't bother about the technical details. But this was enormously surprising that what we saw is that for the semi-terrestrial clades is this freshwater clade. There is a brackish water ancestor. A brackish water ancestor which became semi-terrestrial. And then first the semi-terrestrial or the terrestrial habitat was colonized, and then they went to the freshwater. So this brackish water inhabitant colonized the terrestrial habitat. And then there were at least 2 independent colonization of fresh water out of the terrestrial realm, which was completely the opposite of the last 150 hypothesis of the last 150 years. And this is some more detail of one family where you see that even within one smaller group there are many different freshwater colonizations, but I'm not going to detail. This would lead us too far. So we know a lot about, well, not a lot, we know a little bit about habitat shifts now based on phylogeny. This were 2 PhD projects. The third one I had last year went on this Giratrix hermaphroditus species complex, which is Giratrix hermaphroditus here, species described from basically everywhere, you will see, and which is characterized by a stylet which runs through a sheath with a stalk. So this is the copulatory organ basically. And, well, sideways it uses this for copulation, we filmed it, but it also uses for stabbing prey. So it's used in two different ways. Strange thing is when you look at the distribution, the world distribution of Giratrix hermaphroditus, it occurs everywhere, and in green is freshwater and in red is marine. So it occurs in all types of habitats, everywhere. And now this is very strange. I found it everywhere, in sands, in algae, in freshwater, in marine habitats. There is one coast where it doesn't occur, it's the Belgian coast. Never saw it here. Don't know why, but it doesn't occur here. And it stops somewhere in the German Wadden Sea and starts again occurring more or less around Wimereux, Boulogne-sur-Mer. So that's doesn't like Belgian beer, I guess. It's very strange. But for the rest, you find it basically everywhere. And then this occurs is this dispersal, is this over-conservative taxonomy. Well, in first instance, there are arguments for the latter, because few studied the chromosome number, and then already it's seen even in Australia, you see differences in chromosome number. So this is quite a good indication that they're not the same species everywhere. And then we started studying it. A student of mine did it with based on several genes and then several automatic species delineation methods. I'm not going into details either. But what we found is that depending on the method used, there are about between 63 and 75 species in this species complex worldwide. And the rarification curve showed that we didn't find all yet. So there's an enormous number with a freshwater clade again and a marine clade. So here it's pretty obvious that it once came from, well, in one time came from the marine realm and invaded the freshwater once. But the strange thing is they occur everywhere, all continents. Even when you have, and then we were able to link these putative species to, well, these clades that came out of the molecular analysis to small morphological differences, yet very consistent differences. So now we are able to distinguish all these species based on morphology alone. And these are all the morphotypes we have. And within the morphotypes, different species can even be distinguished by morphometric data. And the success rate of identification is really, really high. So we are now able to distinguish them based on morphology and morphometric data without any problem. The strange thing is when you see this type of species split up in many small species, what do you expect is that they are all geographically localized species, and that they are, which is not the case. Many of these species we found within the Giratrix hermaphroditus complex still have an enormous distribution over 2 or 3 continents, which is biogeographically very, very difficult to explain basically. Why does this happen? And we are plunging much more into this now, but it's not easy to answer. This, basically is what we are investigating now still further. And the only thing that rests me is thank you. And these, of course, thank all the people who did it. All the results I showed was the work of these 3 PhD students with technical assistance of these 2 people. One who disappeared, basically. I don't know why. Okay, thank you very much.